Hi all, so this is the walkthrough for the week 7 lab on genetic inheritance. You're going to use your lab kit manual and the lab document. You complete the document, make sure everything's in it, and make sure you submit everything to the assignment files tab. Make sure everything's done before the end of the last day of the class, because remember there is no late period in week 7. You won't see the faculty materials, but you'll see the student materials, and you'll have your lab document, the report, and then of course the playlist that you're probably watching right now. So let's go up to that. Let's go up to the rubric, not the lab doc, rubric first. Okay, so here we were going to have some tables. Why they're called tables and not observations, I don't know. We may have changed it. We'll go see when we look at the lab document. And then there's going to be 3 exercises and half a point for grammar. Okay, let me open that. Right, so here's the lab document. Okay, your title page here. So your title, name, course number, so you know Bio 350. That's what we're in, right? I've been doing a lot of these videos lately, and then my name is optional. Because I haven't lost my voice yet. So then this is normal stuff. Okay, so now we have tables instead of observations, but they're basically the same. I guess they don't count as observations because most of this is stuff that you're just kind of filling out. So you're going to do a lovely Punnett square for this doohickey. Make sure when you do it, do your hybrids, that it doesn't autocorrect any recessive alleles to be dominant alleles, because sadly that happens. And make sure you fill in, oh, I should put that in yellow too, that you fill in those blanks. It's thinking about it. That you fill in the blanks for which the genes are. And then that's supposed to be an arrow over that way, and then an arrow that way. It's not too picky, they're like the same. Okay, so then we're going to have an actual observation. So that's going to be how many green plants you had, how many white plants, total plants. You can do it for a couple petri dishes, just one is fine. Okay, then you're going to fill in the blanks over here. So let me scroll down. So what's the expected alleles, what's your progeny going to look like, and then what is the frequency? These are more like arrows. Bloop, bloop. Okay? And it gives you the lowercase one, right? The all recessive. So then in here you're going to get to do a nice cross for two different traits, right? For your F1s. And you will put in, just checking that has everything. Yeah. You'll put in over here what your frequency is, and what's over here is your frequency. And I guess I could make these yellow too, as long as we're being consistent. Okay. And then here, well, this one's blank, but up here you'll put, your different options. Here are your different options, and then fill it in. Your most dominant should be up here. Your most recessive should be down here. And it says student fill in all, so filling it all in, whatever needs to be filled in. Okay. We go to the table. So we're going to put in the colors here. Then we're going to say what are the different genotypes. I don't know why it says genetic designations for a phenotype. So the various genotypes that could be responsible for these phenotypes. Oh, I don't like that. Let's hit save. Okay, and so they give you an example that your full recessive will be yellow and wrinkled, right? And then you're going to have your expected ratio. You're looking at 2 traits. So what is that? Write it as a decimal, right? And then you're going to have, up here you're going to have the actual observed number. Put the total number here of however many you had. So if you had 100, 100 will go in here. If you had 123, it will go here. And then this will be, you saw this many, this many, this many, this many. Then the actual allelic frequency. So it will be whatever the number you saw divided by the total. So if it's 100, it's really easy because you put like 53 here. 53 divided by 100 is 0.53. But if it's a different number, you'll just divide. So it's no big deal. And then things are talking to me. And then let's go down to the next one. So now we're going to do our chi-squared. Pretend it looks more like a chi-squared than just a big X. And in here you're going to put down first the different colors, which should be the same ones that you used up here, right? So let's go plunk over here. You're going to have, again, from here, week 4 table, you will have your, which one is first? Your observed, let's just do a split screen. So go to view, split. Thank you. Can't see the top. Alright, so that gives me the top of table 4, right? Let's go a little higher. And let's look at table 5. So observed number from table 4. So this is going to be the observed number that you saw. So that goes plunked over here. Observed ratio, plunked over here. Expected ratio, it's going to go plunked down here. Expected number calculated. So that would be if I had expected number, so if I'm using it at 100, it's really easy. If I'm doing it like 123, I'll have to take my expected number, multiply it by 123 to get my expected ratio. Multiply it by 123 to get my expected number. If it's out of 100, you just plunk it over, right? Nice and easy. But if it's more, you just make sure you do that. And then this one, which I'm sure I'll tell you right underneath. Let's look and see. That'll probably be a good thing to check. It's not there. Okay, and then that we don't need a split view. So let's remove the split for a second, make sure. Yeah, so the actual allelic frequency observed ratio is which one? Oh, no, that's not that one. This is this one. Okay, so the expected number, it needs to be it does need to be a double. I think I gave it, changed me over, and I let it. Don't let things change. Now let's go. Undo automatic bullets. So this one needs to be doo-doo, right? And then this one instead should be 1, 2, 3. There we go. Let's go space. Alright, so expected number is going to be the sum of the observed number times the expected ratio. So the total of whatever, like I said, so how many you had multiplied by this. Then we have the observed number minus the expected number squared minus expected, divided by expected number. So it's observed number minus expected number squared, put D in there, divided by expected number. So let's say the observed number was 5, the expected number was 4, that'd be 1. 1 times 1 is still 1, divided by the expected number of 4 would make it 0.25. Okay, because 1/4 is like 1/4, because it's a quarter. And then chi-squared is our triple Q, or our double Q, C, blah, blah, blah. See how it does C here and it does an X here? We'll just, whatever. Just accept the fact that things are inconsistent. And whatever here. So it's going to be the sum of all of these suckers together. That's... Yeah, so you're going to take the sum of all of these buddies together. Let me put another parenthesis just to make it right there. Oops, wrong one. Did I just hit number lock? Yes, don't hit number lock. Okay, there you go. So all of those values. So it'll be all of these guys in this column. Like let's say I have 0.25. 1, 2, 3. So 1, 2, 3 would be 6.25. That would be my chi-squared number. Yours should end up being around 2.1-ish. Okay. So then you're going to put in whatever your chi-squared was. You're going to value at 3 degrees of freedom that is closest to your chi-squared. You're going to say what is the fit probability. Okay, so what was that number that's the closest? What does that put it in? What's the percent of that probability? So if it's like 0.5, be 50%. And is that a good fit or a poor fit? And you'll just delete the one that it's not, right? So then we go to exercise 1. So then what's the expected ratio of green to white? So the expected one is what to what? And then you can do the calculation of the expected ratio, blah, blah, blah. Just give it to me in decimals. Here whole number, here decimals. That is the easiest way of thinking about it. So then, if I know I have 320, how many green do I have? So we're looking at this decimal up here. I have to multiply those suckers together, and you get here. And then you'll do it for the recessive one, same thing, right? So then when you look at the hypothesis, if it's good, how similar are the observed to the expected results? You want to give me your numbers, what it should have been numbers, how close are those? If the results are not similar, how might the differences be explained? Hypothesis, even if they were actually the same. will the monohybrid F1 cross in the corn yield to the same ratio of expected phenotype and progeny as for the tobacco seedlings? So long as they're hybrids, does it make a difference? I mean, monohybrids, right? Does it make a difference what it is? Assuming there's independent assortment, right? If available, compare your F2 seedling data. That's the space. Let's check and make sure that is a space, not a random period. Isn't that weird? It like moves. Whatever. Okay. Are the outcome ratios the same? Might the number of seedlings might a larger number of seedlings basically be better? Where's E? So basically, is a larger number of seedlings a better idea? Why? If you flip a coin once or twice, how that gives you what observed ratio versus if you flip it like 200 times. So based on what you conclude in the genetic makeup when told that the corn plant cross P, pictured in figure 2, is between a completely dominant and a completely recessive plant. A, construct and record a hypothesis of what the F1 progeny of the dihybrid cross will be. So these guys, you cross them, what are their babies going to be? And then 2, you take those babies and you cross those suckers, and what do you get? You can stick in numbers, what the genotypes are, whether or not they're hybrids, that kind of thing. So then we have, what are the 2 hypotheses that you made about the allelic frequencies by the process? So P dash P, what are they going to be? What are they going to look like? And F by, I said P dash P, P cross P. And F1 cross F1, what are those frequencies that we're expecting? We're looking at 2 traits. What's the frequency? So one genotype is going to be completely what? And the other one will be, that's recessive. The dominant one's going to be what? And completely recessive will be what? Does it make a difference instead of having one completely dominant and recessive if one's completely dominant for one, recessive for the other, and then it's reversed? From the phenotypes of the corn, oh gosh, I'm taking a while. I just looked at how much time I left. We need to go faster. Okay. Let's go faster, my dears. Okay, the phenotypes for the pecan would be what? You can figure that out. Okay, so when you do this one, it's 2 to the nth. So you figure out what your n is, and you're going to raise 2 to that. So if I give you an example, 2 to the 3rd would be 2 cubed, or 2 times 2 times 2. So you have to figure out what your n is. Some people think the answer is just the n. It's not. And then if the F2 corn crossing results in F1, blah, blah, blah. So we need to know our things were F1, right? So F1, does that mean we use our 3 to 1s, or do we use our 9 3 3 1s? Which ratio are you going to use? Dihybrid, you should be okay. Applications that you might use this for medically. I'm still going to run out of time. Okay, I can do this. Okay, blah, 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 outcome. So the big one here, how close is it to observed? That's fine. Tell me, ignore this little one here, it just showed up. You need to tell me what numbers, like what results would give me a good fit and a poor fit. What does this mean? Like if I say it's a good fit, is that a good study? Why do I care? Why do I care what fit means? Like what does it mean? So let me know. A typical cross where the dominant, you figure that out. You can figure that out. Give an example. Okay, so this one, we're dealing with that F2. So in F2, are we looking at 1 or 2? Has extra space. 1 or 2 traits. So if we're looking at 1 trait, we have what kind? And 2 traits, we have what kind of ratios? Make sure you check that. Okay. So if all our stuff is doing like things like they should, does it matter? Will everything follow Mendel's, right? If things are weird, are we ignoring some of Mendel's laws, right? This should be this should be a question mark. Don't you like how I change things? Okay. So we have 15 seconds, so you're going to do your lab summary and you'll be done. Yay! Just Send me that draft by Monday at noon. I hope this helps.